Pleasure to introduce Kat Dawson as the Colloquium speaker for our seminar today. So Kat originally grew up in Philadelphia, not too far from here, and she attended Penn State to do her PhD. She worked on her PhD thesis under the supervision of Kate Freeman and Jen McElhatty. After she finished her PhD, she moved out to California to work with Victoria Orphan at Caltech. And she spent 6 years as a postdoc in the Division of Geological and Planetary Sciences at Caltech before she moved back to the East Coast and joined her faculty here at Rutgers. Now, despite her training in geological and earth and planetary sciences, her research is very biological. As you'll find out, the programs in geoscience and planetary science at Penn State and at Caltech have much more focus on microbiology and biological sciences, and she integrates these tools in her cutting-edge research program. So in my opinion, the type of work that Kat's doing right now represents the future of low-temperature geochemistry and aqueous geochemistry. So Kat's going to talk about microbial methane metabolism today. Take it away, Kat.
No pressure. Being the future here. Yeah, so I changed my title slightly. Oh, lights. Okay.
Press one of these buttons. Is this better?
Cool. I changed my title slightly to specify which tidal river I'm going to be presenting results on to being our own local laboratory, the Raritan. And this talk is going to take us from some of my postdoc work in the deep sea to work done by students with me this summer on the river here. And, oh, I just did not do that correctly. All right. Forward. Oh, I'm just going to press. Okay. And so specifically in terms of the deep sea, I'm going to be talking about the Santa Monica Basin and some methane cold seeps that occur off of the coast of California, but within view of the Santa Monica Pier and the iconic Ferris wheel. And then move on to talking about this natural salinity gradient and how it presents us with an interesting observatory system here in New Jersey. So first to talk about Santa Monica. So when we're talking about methane in the deep sea, primarily we're talking about very ancient carbon, but much of it may be relatively recently generated, and this can be both by thermogenic processes as well as biogenic processes, where various organic material that's come off the continental shelves gets broken down eventually into methane, which then can percolate up from hydrates to the seafloor. And you see these sort of interesting and very common distributions around places where methane is bubbling up onto the seafloor, where you have an initial zone of very high methane concentration that can be deeply sulfitic, or very, very highly sulfitic, as a result of chemosynthetic bacteria and sulfate-reducing bacteria utilizing sulfate from the overlying water in accompaniment with anaerobic methane-oxidizing archaea in the sediments. And typically you see these sorts of profiles here where methane coming up drops down sharply, sulfate coming from above meets it at some transition zone point. And we also see very, very depleted delta 13C signatures for DIC associated with this, and often carbonate precipitation. Now, of course, as you move out from this zone of initially very high methane concentration and very high sulfide, you start to see this sort of oasis effect where this carbon inputted into the seafloor, as well as the sulfide, represents a really fantastic energy source for a diverse set of both micro and macro communities down there on the seafloor. So we see tube worms, clams, and mussels, as well as many other larger organisms out there. But for my sake, I'm interested in usually digging down and looking at the microscopic level. And so this particular aggregate of bacteria and archaeal cells is about 30 or 40 microns across. Is that true? No, it might be a little bit bigger than that. But these are very, very small scale things within those sediments. So in 2013, I had the good fortune to go out with the folks at the Monterey Bay Area Research Institute on their research vessel, the Western Flyer. And we went off of the coast of California, starting in Monterey, but eventually working our way down to Santa Monica, and utilized their remote operated vehicle, Doc Ricketts, which has these pneumatic arms and capability of taking push cores while we sit comfortably and have cups of coffee in a control room. And it's a great way to get an awful lot of research done very quickly while getting fantastic high-resolution pictures of what you're doing at the same time. And in this particular mission, we chose to try and investigate differences between the chemical distributions and the geochemical distribution within the sediments, the overlying surface community composition, as well as integrating some novel investigative techniques involving environmental proteomics and sulfur isotopes at the same time. And so using these sort of disparate data sets, this is an effort to bring them together into a way that makes a more cohesive story about what's going on in some of these settings. And so although we went all the way down basically to San Diego, the real focus of this is going to be basically one little seamound that we took an awful lot of push cores in, as well as a couple more that were slightly higher up the slope towards Santa Monica. And so these what is that? I believe that's 10 sites, included 21 cores, and in terms of individual sections, 130 individual samples that were sectioned and analyzed here. And so what do these different habitats look like? So I showed you the cartoon image before, and so in these high-resolution images from the ROV, what does it actually look like? Well, we have these clam beds where you can see, you know, a very dense packing of clams, and I'm sure there's some mussels in there too, to bare seafloor as another end member, and then mats of orange, white, and sometimes occasionally what appears to us as a bit of a pinkish color that can have a variety of textures, from very, very spongy to basically like cotton ball fluff that would float away at the slightest pass of motion over them. And what else you're seeing here is some of our other instrumentation that went down for this particular cruise. The tripod thing, unfortunately, didn't work while we were down there, although it would have been lovely if it did. Was meant to be getting in situ methane measurements. These over here, these sticks covered with what appears to be black smudges, are actually black and white photographic films. Just absolutely that, the kind of thing you could go to a camera shop anywhere and purchase. And they've been cut and fitted onto hard plastic stakes that can be pushed into the seafloor. And so the great thing about black and white photographic film is that it is actually, it's coated with silver nitrate, and silver reacts very readily with sulfide when it encounters it, and in very, very short time scales. So within 15 minutes of placing one of these films into this sort of sediment, or sediment in perhaps the pond over on Cook campus, you can see these black stainings in reaction to the in situ sulfide concentration. So we can pull this out and within 15 minutes have a picture about where things are going on. And better still, when we bring it back to the lab, we can cut it up and we can actually extract the sulfide off of there for isotope measurements. Or if we're being really excessive about this, go take it to a nanosims and raster across and get very, very high resolution profiles of the sulfur isotopes from an in situ capture of the sulfide. And so that's one of the exciting things that's come out, and a lot of that has really been out of Dave Fike's group at WashU in St. Louis. Right, so what we're doing with these push cores once they come out of the seafloor, and they're generally in this kind of range of 20 to maybe 25 centimeters total. Occasionally you'll see a nice big chunk of microbial mat sitting at the top of them. Otherwise, sometimes a clam might be sitting at the top, depending on what we took. Is we're cutting these into very fine segments. Initially at the top, we were doing 1 or 2 centimeter resolution. and then deeper down where they anticipate it, once we anticipate we're past that sulfate methane transition zone, we're doing slightly bigger sections. The sediment then goes into a Reeburgh-style squeezer, and so this is using an overpressure of nitrogen gas or argon gas at the top to force water out of the sediment and into these various syringes so that we can then do what we choose to it. But by doing it this way, we're attempting to keep it minimally exposed to oxygen throughout the entire process in an effort to preserve various sulfur species. And from this, from any given sediment horizon, you can typically get anywhere between 5 and maybe 20 milliliters of water, depending on how thick the horizon is and also how densely packed and hydrated it might be. And so that's a lot of water, or a little bit of water, depending on how you think about it, and what you choose to do with it, and how finely you can divide that up limits your future chemical analyses. Okay, so in terms of a seep habitat comparison, this is all of the chemical data that we took from there, and this is a little bit too hard to read and everything, but one of the things that just at first glance you should see, and so any given site will be going across this way, and the individual chemical parameters are going from top to bottom, are going downwards. And so just to zoom in a little bit on this, we can focus on some of the big ones, the delta 13C of the DIC concentration, sulfate, sulfide, nitrate, and ammonia, as well as the sulfur isotope profiles. And so the cool things that come out of this is that there's kind of a very generic profile that I think we all know from literature in terms of these settings at some point in time, having come across it, that you'll see the carbon isotopes go down to basically where the methane carbon isotope value was initially. At the same time, sulfate goes from perhaps near seawater concentrations to near zero. Sulfide, it has the opposite directional effect here. Anytime you're seeing this, that's because we weren't fast enough and we lost sulfide by degassing because we were too slow. And it's something we're constantly working to get better at. And then, of course, the sulfide isotope profiles, because we're so rapidly using sulfate in this system, are all going to basically about 20, 21, 22 per mil for delta 34S, which is about what seawater is. So in these sort of systems where there's just an incredible demand for sulfate, it's going to basically just be distilled right to that point. And then otherwise, it's the kind of profiles where we were expecting there to be maybe slightly more differences between different surface environment textures, and really weren't seeing it. And you know, that's okay. But that just means we have to pore through this a little bit more to figure out where the differences are. I think that's twice. And likewise, when you come to the sequence data, because for every single one of these 130 samples, we also went ahead, extracted the DNA, and sequenced them. And this is the picture of the most abundant things, or things that have at least 1% abundance in any given sample. And of course, this is also very hard to read. And so we can go ahead and zoom in just a little bit to the things that are actually the most abundant, and separate this out to just pick one of each type of environment. And what you can see is that there's certain taxa that appear in these environments that seem to be sort of ubiquitous. It doesn't matter if we're looking at a background setting, a clam bed, or whatever. These various planktomycetes and chloroflexi groups seem to be everywhere. And I'll tell you that from what I've sequenced and what I've analyzed of other samples, this also appears to be true in seafloor sediments. There's certain groups that either by bias of our techniques or by just the fact that they are so ubiquitous will always be there. But there's other groups that we can see have differences in abundances based upon the depth of the sample. And so as we move from left to right across any one of these colored columns, you're getting deeper. And so you can see that, for instance, this ANME1, one of our anaerobic methane oxidizing groups, likes to be apparently at these very deep depths. whereas there might be other ones, there's an adenine 2C, that may be slightly more abundant in the shallower sections of our core. And now there does appear to be some differences in distribution between our different sites where the methane oxidizing archaea aren't really that present in our clam bed or our background settings. Right. So then in terms of our sulfur isotope profiles, going across all of these various samples that had been in place for between 15 minutes and a couple of hours in the sediment, what we're seeing in general is that as you go deeper in depth, you come closer towards 20 per mil values or seawater values for sulfur isotopes. But you can see that there's a bunch of different lines in here, and that's sort of getting into the fact that the rate at which the sulfate appears to be being reduced as you go down in the sediment is related to the environment. A location that may have clams that are digging into the sediment a little bit and injecting maybe some overlying oxygenated waters or more sulfate deeper is going to push this profile to come to that 20 much later. And then the other thing that we noticed as things went down, as we processed this data, is that depth may not necessarily be the best way to be comparing core to core. And in this sort of system where everything is really driven by methane and sulfate, considering the fraction of those very important reactants in here and the fraction of what is remaining, it may be a better way to do cross-core comparison. Among other things, it eliminates any time that someone, that the pneumatic arm pushed the core in slightly too far, and you may not know that zero is exactly the same across cores. And so this may help us with that. And so this is looking at the delta 34S as a relation to the fraction of sulfate remaining. And yes, there are some points scattered off of this, and that may be due to some reoxidation of the sulfide in these sediments. And then finally, in terms of the cool data that we were generating here, we used this technique of dual deuterium and 18O labeling to get at quantitative analysis of the environmental proteome. So this is taking the sediment, freezing it, and then not in my hands, but in the hands of Jake Wallbauer at the University of Chicago, doing a bunch of these techniques to get the proteomic data. And what we're seeing here is the distribution of different kind of classes of proteins within these settings, and that would be, things associated with carbon metabolism, things associated with nitrogen, sulfur, etc. And then just focusing on the carbon, sulfur, and nitrogen, you can see that there is a peak in the sulfur metabolizing proteins that correlates with roughly with a peak in the methane metabolizing proteins, and so things appear as we would expect them to appear. But this also lets us dig deeper and see specifically, with some amount of phylogenetic specificity too, who might be responsible for different bits of this process at different sediment depth zones. And then by contrast, you can see that things get a little bit more, a little bit less to the sulfate reduction and methane metabolism in the clam bed, and a lot more other carbon related processes going on there. And I think, interestingly, in terms of a nitrogen story in the deep sea, which I don't think that we often spend a lot of time focusing on in the sediments, there looks like there's a lot of nitrite reduction going on, which is something that we're hoping to pursue further. So what can we do to integrate these things to get into the heterogeneity within methane seeps? And so just looking at basic correlations, and so this is taking Pearson correlations between various parameters, and you can extend this matrix out obviously quite a bit further, but I'm just going to zoom in on the geochemistry and the microbial diversity. Again, what we're seeing is that depth is this really impressive driver in this system, and of course it is, because methane comes from below and sulfate comes from above. So lots of things like sulfate concentration and sulfide concentration are going to be very distinctly related to depth. Again, to why we're trying to remove depth as a driving factor here. And that there's lots of different taxa that were relatively abundant, and that seem to have different depth distributions. And so this I'll show you in a little bit later, taking this into network form and trying to figure out how these different groupings may be interacting at different spots. But first, getting back to this fraction of sulfate instead, we can start to kind of clean up these sulfur isotope profiles by using that as opposed to depth as our x-axis, and then also plot on the abundances of various taxa here. And so as I was saying, depth seems to be this thing that really drives things. And so if we just take that away and look at the fraction of sulfate, now we start to see that there may be different zones and different amounts of sulfate that are associated with different sulfate-reducing populations within this system. And this sort of information helps us get potentially at who partnerships in terms of a microbial world might be, but also at who is responsible for imparting certain bits of these sulfur isotope signatures onto the DIC and potentially carbonates that are precipitated out here. And so in particular, we're seeing that the yellow, which are our Seep SRB2 group, appear to really be abundant at very, very low sulfate concentrations, while there are these other ones, including the SRB1 and the disulfo bulbous group, that appear to be more at the earlier stages. We can also do a similar thing with the DIC concentrations, and then I'm just throwing on here the fraction of sulfate remaining too to show you that there looks like there's this typical uptick at the end at a certain point when there's very low sulfate, maybe suggesting fermentation. And so what we're seeing here is that there's also these different populations of our methane oxidizing species that pop up at different zones, with a set that prefer to be in this low sulfate zone and a set that prefer to be in the higher sulfate zone. Right, so in a network sense, and the names are not necessarily important here. The important thing is that when we take a look at all those correlations from the sediments, we basically get three broad clusters. And the interesting thing here is that they're actually totally depth-related clusters. We have this sort of mid-depth methane oxidizing cluster, a deep depth that looks like it's very much related to ammonium and nitrogen processing, and then this shallow heterotrophic mix of everything depth of microbes from these sediments. And in terms of just that methane cluster, there's some potential new friends that we haven't really assessed often that are worth potentially looking more in a microscopy sense into, including this Hyde 2412, which now has some candidate phylum name that I can't quite remember, as well as this potential association of the ANMI1A and ANMI2A2B group with this disulfobulbous sulfate reducer, as opposed to the most abundant one that typically is in our sediments, this one here. And so this is getting at that maybe slight niche differentiation between our populations within our sediment. And so we can further support some of that evidence of relationships that we hadn't quite gone into before by combining our proteome data with our sequence abundance data from these settings too. And of course, everything here is based on abundances, and that obviously means that at some point we can make predictions here that need to be further explored. But they're useful predictions that seem to keep occurring no matter how we look at things, and just the geochemical parameters, just the protein parameters, just the abundance parameters. And so that's valuable support to the fact that this is something worth pursuing further. And in this case, we see when we look at our taxa abundance with the proteins that our Seep SRB2 and our 1B groups, so one of these ANMI1s, again appear to be really closely linked together and linked with a bunch of nitrogen-related genes. And so that's interesting from the sake of being a system that's so dominated by sulfate and methane that now we have this maybe peek into the fact that there's this nitrogen and nitrite reduction thing that may be going on and could be a driving factor at depth within these sediments. And then, there's many other relationships to also explore here. Right, so in summary of this beginning bit, so there are compositional differences between cores at different geographic locations, so site A versus site H. And those appear to be more significant than the differences between cores that have different overlying features at the same spot. And this normalizing to the fraction of sulfate remaining gives us a lot of insight into differences in relationships between our geochemistry and specific taxa within specific cores. And there's a bunch of new relationships that may be popping out here that are worthy of further enrichment culturing experiments and maybe further experiments in the field. And so that's kind of where I'm leaving things on the deep sea for right now. But the truth is that I sort of have this. I had this while where I had changed my home Wi-Fi password to no more ANME for now, and little did I know that I would choose to go and do a bunch of work on the Raritan River and find myself totally proved incorrect about that. So now we're going to move to this, talking about the Raritan River and find out what's going on with the methane cycle here now. And so in June, we went from about Sayreville up to basically New Brunswick, and so deeper water to shallower water here. With the overlying question of what's increasing seawater intrusion into these sort of tidal rivers going to have as an impact on the microbial community and metabolism in these sort of settings. And one of my big reasons for being interested in this topic is that wetlands, if you consider net primary productivity relative to spatial area, are incredibly important in terms of coastal carbon and the carbon cycle in coastal environments. And knowing this, we need to be filling in and getting more details about what's going on in these very dynamic systems that are going to be very dramatically impacted as sea level rises. And if this is a 3-meter sea level rise, so it's not going to be the near-term future, but you can see that the Raritan River is going to be salty quite a bit further up than it currently is. So what's going to happen to net primary productivity and what's going to happen to the dynamic metabolisms going on in these sediments? And so this summer, my group sought to start to do some preliminary investigations of what's going on right now. Okay, so starting with the RV Raritan, we came upriver, sampling right past the train swing bridge, where we're not quite seawater salt levels, but pretty high. Then moving up towards some of these salt marsh settings around Crab Island, and then finally winding our way up to the mouth of Lawrence Brook, which is about the last place where we found decent sediment that could be gathered without just hitting rocky bottom and not being able to pick up any mud. And so all this is being done with basically a lake coring system. So I've got a 40, 50 centimeter core with about a 3-meter pole that we can put into the into the sediment, and so that limits us as to how deep we can go. But we can sample most of the Raritan, especially if the tide isn't super high. And so what we're pulling out are cores that, in some cases, are filled to the top. That can then be taken back to the lab, cut into sections, and processed in a similar way to what I was doing with the deep sea cores. The biggest difference here is that obviously we're doing this hanging off the side of a boat and pushing a core in. So my control over exactly how far I push the core in is a lot less than watching on a video camera comfortably in a lab, comfortably in a video room. And so in most cases, I'm not super confident that centimeter 0 is really the top, and that's something that going to take some work and some practice to get a handle on. And given the turbidity of the river, may never be something that we can do. So what we're doing is cutting these cores into basically 1 to 5 centimeter increments. And each of those increments is getting divided up in much the same way as the deep sea ones for different purposes. So we're sampling for DNA and RNA for microscopy purposes, which I'm often going to abbreviate as FISH for fluorescence in situ hybridization, just to you who's there. And then we're taking extra sediment, spinning it down, and taking the water off of the top, filtering it, and dividing it for various analyses. And so right now, what we're focusing on is sulfide concentrations, iron concentrations, and the various anion profiles that we can get. And these are things that I'd like to expand as time goes by. I'd love to have a DIC profile for these sediments too, and a better handle on what's going on with changes in the pH in them, but in the future. So what do things look like? So as we go through our Raritan sediment, and so in all cases, site 1, which is the saltiest setting, are going to be green here. Site 2, the middle salinity, is going to be red, and site 3, which is basically fresh water, is green. The extra circles on here are the overlying water above these sites, and so you'll see in some cases the overlying water has more of a parameter, and in some cases it has less or basically none. And so what we see in all cases is that sulfate decreases with depth. And in some cases, like our salt marsh setting, we started out at the very top of our core with significantly less sulfate than the overlying water. And this may be a question of how deeply was zero actually. And unfortunately, we were only able to get a very shallow core in the freshwater setting, and so we don't have much idea about what's going on any deeper there. At the same time, we can see that there's a lot of variance in the iron 2, so the reduced iron component that's dissolved into these pore waters. So there's spikes in places, and this is associated with the pore water. There is essentially no measurable iron 2 in our overlying water. This is pore water associated only. And there are other things like nitrate and acetate that are there and that have these spikes within them, suggesting that they're probably associated with processes going on deeper in our sediment. Oh, this is so funny. It's duplicated things a couple times. So what we ended up doing with my lab group is we took the sediment combined and mixed into a slurry and then distributed into bottles and exposed mud from our freshwater site to water from each of the 2 saltier water sites, and including a sample that John's group provided us from the bay. So we had water from the bay that we could mix in with all of these samples too. So the idea was to see what happens to these different sediment communities if you change what their water looks like. And I don't know if the really exciting, interesting, or slightly depressing thing is that over the course of my summer students project, nothing really happened. and sulfate basically, so we're talking about basically the first 30, 40 days of the experiment, sulfate did nothing. We lost no sulfate in any of these experiments that we set up. And to me, I was just blown away by this. Why were we not losing any sulfate? You had these clear evidence of sulfate reduction going on in our sediment profiles, and I've now taken mud that looks like it's lost all of its sulfate and given it some, and nothing happens. Well, it seems like what the real issue is that there is so much iron 3 associated with humic material or other mineral fractions in these sediments that we have basically a bit of iron buffering in all of our sediments before sulfate reduction can really even take a hit on anything, and so it would appear that it takes nearly 60 days of exposure to higher sulfate water before you start to actually see sulfate reduction going on. And so this is an interesting thing to be noting from the perspective of having high storms and intrusions of saltier water and more sulfate-rich water upriver, like how long before we start to see shifts? And it may be 2 months or so. But that 2 months may change as things go on, and how much iron is actually present in our sediments to continue doing this buffering effect with time. And those are questions that I think remain totally unresolved at this point. But basically one of the things that we do note is that there definitely does appear to be greater iron reduction and greater iron 2 production in sediments that were exposed to higher sulfate water. So there does appear to be a link between those two, and that the iron concentration at the start appears to be greatest in our Site 3, our freshwater setting, and progressively less as we get to the saltier water setting, suggesting perhaps that the longer time this sediment has been exposed to sulfate-rich water, the less residual buffering capacity of iron for sulfitic sediments that there may be. And there's definitely the appearance of blacker bands in here, suggesting that there's likely some sort of iron-sulfur precipitates that are collecting. Right. So who's doing it? Again, we went ahead and sequenced everything, and so my lab is currently set up for doing DNA extractions and tag sequencing, including our own set of primers, barcodes. We can pool samples and send them off to the point that it's probably between $20 and $25 a sample to get 30,000 sequences for that sample. And I'm more than happy to collaborate with anybody who's interested in any environment. And again, you're going to end up with just a massive amount of data to sort through and make heads or tails of. So as you're moving across here, again, left is shallow and right is deeper. And every time you hit a shaded zone, that's moving to a new core. And just to zoom in a little bit, what really excited me and kind of astonished me is that there's very abundant anaerobic methane oxidizing archaea in these shallow tidal river sediments. This is not something that I was expecting, and I almost had to sit and go, have I somehow managed to contaminate this? What could I possibly have done that? And everything is newly purchased and has never seen the labs in California, so no, I don't think so. And do I have pictures of or headshots of who these guys are yet? No, but I'm going to work on that. So I think that this is exciting from the perspective of being an environment you wouldn't necessarily anticipate this to be going on. Methane production, yes, and we do have methanogens here also, but they're actually not necessarily as abundant as these methanotrophic archaea. And we can make that even a little bit more clear by putting them in terms of depth. And what seems to be going on, and right, okay, so site 1, 2, 3, and the colors are the same from before here. What seems to be going on, and just to say that this data is from a parallel core, so it's not the exact same core, is that there appears to be these peaks, particularly in our ANMI2A, 2B group, that appear to co-occur with peaks in the iron 2 concentration, suggesting that there may be an iron-based AOM activity going on in these sediments. And while our methanogen populations are here, my suspicion is that they may actually be a little bit deeper down, or they may just be not very active in these locations. Right. So canonical AOM, of course, is methane plus sulfate to bicarbonate and sulfide, and it's very, very low in terms of energetic yield. And so methane and iron hydroxide to iron 2 and bicarb is actually quite a good energy bet. And so it's not an unreasonable expectation. It's more a matter that these organisms are typically so sensitive to oxygen that you wouldn't anticipate them in this sort of location. And in the deep sea, there's not the abundance of iron that would allow this to be the dominant process. So I guess my lingering questions at this point are both iron and sulfate coupled AOM occurring in our river? And how prevalent in general is this in tidal rivers? And also, what role does that play in the coastal carbon budget? Because these guys will be sucking down methane, of course, a more potent greenhouse gas than CO2. but at what rate and what importance is that to anything in general? Right, so finally, in summary, yeah, so sulfate concentrations increase with depth in all sites, and we're seeing these unexpected high abundances of ANME, and it looks like there's a correlation with the iron and the ANME abundances. And that's really it at this point for this story, which requires a lot more work, and fortunately I've got time to do it. And finally, as a bit of like shameless plugging for my lab, I've got some newly arrived exciting instrumentation, and some of it hasn't quite arrived yet, and it will be coming soon. And I'm actively seeking collaborations from anybody who might be interested in playing with these toys with me. And so I have a GCMS with a FID, and the column is split. You can get simultaneous quantification and identification of your compounds, and I'll have it set up for doing either gases, nitrogen, methane, whatever, or with a different column for organic compounds. Soon there will be an epifluorescence microscope, and I've got this with the potential for doing Z-stacking capabilities, so we can do 3D reconstructions to some extent. And finally, and most exciting to me, sometime in the spring, there will be a new isotope ratio mass spectrometer on the Cook campus. And this is Elementar's model, and their pictures. I don't know if it'll look exactly like this. But so what I'm getting is carbon, nitrogen, and sulfur capabilities for the EA, and that'll include 32, 33, and 34S. We'll see how good the 33 is. And then in terms of headspace analysis, and that would be acidification of carbonates, waters, or otherwise headspace, will be set up for doing CO2, N2O, and methane. And I love fun new samples and fun new environments, and I would be happy to work with anybody or anybody's students on projects involving these instruments. And thank you.
Questions for Kat?
So, where is the methane coming from in the Raritan rivers? We don't see much in the way of plankton.
Yeah, so I think that the methane engines that are there are responsible for the methane, but I think that there's another possibility here too, in that but near the Lawrence Brook, there's actually, and I should have stuck this as an extra cheat slide or something like that, but there's a big landfill there, which is actually the largest point source of methane emissions in the state. And I think that there's a possibility that there may be some leakage of that methane into the water system as well as into the atmosphere. And so there may actually be much more methane than we think. But yeah, sorry, I'm just gonna do that. Okay. But yeah, so I think it's a couple. And with this in the future, hopefully with the methane isotopes, we may be able to differentiate between two pools. Yeah.
One comment is that iron is probably partially being delivered from iron-rich clays and a lot of red clays in the Passaic Formation here. And others in this room know a lot more about it than I do, but that's probably, and we see that, when you go to the rocky part where you can't get through the pores, you see that red clay there. The other thing is the nitrate is incredibly high in the water.
Yeah.
And I don't know exactly how you did these incubations, in terms of sediment versus water and stuff like that, but that's another thing that would slow down sulfate production.
Yeah, so in our overlying waters, the nitrate is high, and I'm not necessarily surprised by that because there's enough input from washing off of everything. In our actual sediments in the incubations, by I think we sampled them at 11 days and then at 15 days or something like that. It's basically not detectable by that point, and so it's already got, when I noticed it in the water, I was like, well, we'll see what happens, but I figured that it was going to get used right away.
So what's your strategy for figuring out whether this is like a peculiar thing about the Raritan or this is like
So yeah, so the next place that I'd like to go is down to the Marine and Coastal Science Center on the Molucca, and I think that presents another good representative of a river that's hypothetically less industrial impacted. And I think after that, it's a matter of taking a look at some maps and figuring out good spots that are likely to have some sediment. I think you probably do need at least 20 centimeters of sediment before you're going to see this sort of stuff going on. But I think there's places to pick. And I also noticed that there's some iron carbonate deposits in the Raritan too, aren't there? I don't know. Someone here probably knows better than me. But I was looking at there's an app that's called. There's an app for picturing what's below you, like the star app, but for doing the geologic formations underneath you, which is very cool. And so I'd been staring at that and being like, what is this? So yeah, I think yes, it's gonna start here, but move a couple other rivers soon.
And you also been north into a bunch of chlorine, sulfur environments, and expectations with this starting to get into that. So there might be an opportunity, not rivers this much, but
That would be excellent.
It's safe to see that a lot going on and whether
Yeah. Yeah, well, and I think that's a great setting for a lot of this kind of stuff to be happening in too. So yeah.
Can I ask a naive question? A lot of your profiles, your depth profiles, show a curve of mixing between two endpoints, in my opinion. But what do you see as part of the time frame for the accumulation of, say, your depth? In other words,
Oh, what's the sedimentation rate? I do not know. I assume sedimentation rates are pretty high, but it's also with a storm. I'm guessing that you get a lot of stuff swept out here too. So someone in this department probably knows a lot better than I do.
so if you look at slightly longer profiles, do you see things flatten out and becoming linear, or do you see continuing curve or change through the data?
Well, right now I'm limited by being able to get that 50 centimeters, and also in most cases I'm limited by hitting hard bottom, and so I'd need to change to get a vibrocore or something, I think, to get a bit of a deeper one. And if I can figure out where one is that I can get a hands on for a day or two.
I guess not doing what you're doing, I don't really understand what you're doing, but I'm trying to understand what those curves mean. If they're looking at just a mixture of times between endpoints, or they're really recording something that gets preserved in depth.
Right. So in terms of the Raritan, or in terms of the Raritan. So yes, I think you're looking at you're looking at a mixing of endpoints. An endpoint that has minimal less recharge of water, and an endpoint that's sitting adjacent to the water, so that it is being recharged much more quickly. And so you're seeing that overlying water with whatever nutrients it's containing slowly be used by the sediment microbial community to the point where you're not seeing a refreshing of it on at least the time scales of that sediment's being deposited, I guess. And honestly, I don't know how quickly sediment is deposited in the Raritan, which is something that I should figure out.
I'd be curious if it's very short, or annual, yearly, 100 years, 5 years.
1,000 strikes me as very, very long. It would be too long. I think that this stuff is.
Be on the order of half a centimeter.
Yeah. I think this stuff gets deposited, and then if you have a major storm event, you could wash a lot of stuff away, and then it starts again. Yeah.
Is the Rutgers vessel large enough to deploy a multi-core?
So there's no A-frame on this one, and so I don't know if you would necessarily be able to do it. So basically, it handles about 20 people, although I'd say we had 10, and 10 felt like a lot of people. and so with 10 people, you can lower the back of the boat so that you can basically walk out and touch the water. And if it can be manhandled over the edge and then pulled up with some help from a winch, but if it can be pulled up that way, then yes. But typically when I've worked with multi-cores, we've had an A-frame lowered over the side and drop it, and so.
The
Yeah, yes. Yeah, so my thought next time is that I may go out with a separate pole of some sort, another 3-meter pole that we're gonna note a line on. And you know, when the T-handle hits that line, you stop pushing, because we're at the point where we want to be. And yeah, the trick with these lake cores is that they require some amount of sediment in there to not just flush themselves out too. And so fortunately our clay sediment is very good at sticking inside there. But yeah, it's things that I'm definitely thinking about for the future, about how can we improve this. And yeah, a multi-core would be nice, because then you'd have a bunch at the same time. Yeah. Well, even here, going up to that Crab Island location, and we started at high tide, and definitely they were getting nervous about where they were and getting stuck, because there's nothing more to do on the Raritan when the tide goes too low for your boat than sit and wait. And I've done that with friends and family before, but I don't think the captain of this boat was really going to be happy to do something like that. Honestly, I think a lot of this, especially in the shallow spots, could be done with a kayak or a rowboat too. And so I think there are a lot of possibilities here that may not require renting a boat for a day. But, yeah.
Let's thank